Hello, everyone, and welcome to the Lexogen and Bluebee Scientific Customer Webinar. I'm Ekaterina Alexeyeva, a Senior Marketing Manager from Lexogen, and I'll be a moderator for today's event. This webinar will take about 1 hour, including 3 talks and a question and answer session in the end. Before we start, I want to introduce you to Lexogen and Bluebee. Lexogen is a transcriptomics and next-generation sequencing company focusing on the development of technologies for complete transcriptome sequencing. Its portfolio includes innovative molecular biology kits, software, and services for RNA-seq. Bluebee offers a secure global bioinformatics platform to process, analyze, share, and store genomics data. Through a secured cloud service, Bluebee supports users in clinical, diagnostics, therapeutics, and research with advanced analytics for genomic data-driven medicine. Bluebee offers its partners diagnostic and translational research assay kit providers fit-for-purpose data solutions. And now let me introduce our agenda for today. So the first short talk will be by Lexogen application scientist Alex Moher. He will give us an overview of Lexogen Quantseek 3' and mRNA-seq technology. In the second short talk, the Bluebee field application scientist Andreas Klingenhoff will give an overview of the Lexogen Bluebee sample to data mRNA analysis solution, including the latest addition to it, data analysis pipeline for Quantseek data with unique molecular identifiers. And finally, Dr. Siobhan Gupta, who is a postdoctoral scholar at Dr. David Folt's lab at the University of California, Riverside, will give a scientific talk, TDCIPP exposure during early-blastula alters the normal trajectory of zebrafish development. In this study, he used QuantSeek and the Bluebee platform for data analysis. And now I would like to make an important announcement. This webinar is designed to be interactive, and we encourage you to ask questions during this event. You can submit your questions by typing them in the questions box, which can be found on the right side of the webinar platform. We'll try to answer as many questions as we can at the end of the talks. And now I will turn it over to Alex Moher for the first short talk.
Thank you for that introduction, Ekaterina, and I am looking forward to hearing the talks today. As Ekaterina mentioned, my name is Alex Moyer. I'm an application scientist with Lexogen, and I will be giving a brief overview of our QuantSeek pipeline. QuantSeek is a gene expression profiling 3' mRNA library prep kit. This is an overview of the physical nature of the kit. It comes with all reagents included and is designed for profiling gene expression of polyadenylated mRNA. So in whole transcriptome sequencing, there's a lot of computational resources used and assembly being done, whereas with Lexogen's 3' mRNA-seq kit, we are simply counting transcripts after using 1 fragment per transcript. This enables us to get gene expression profiling data with only 1/10 of the sequence reads needed, enabling much higher level of multiplexing. And on the data analysis side, this requires significantly less computational resources. QuantSeek is, as I mentioned, designed for polyadenylated RNA. The library prep starts with an oligo DT primer for first strand synthesis, followed by removal of the RNA template. Second strand synthesis is done by random priming, followed by a purification step of the double-stranded cDNA library. The final step is library amplification, at which point you have the option to add single or dual indices for sequencing. A new product that we have recently released is the UMI, or Unique Molecular Identifier. What this enables is pre-PCR tagging of your transcripts to deduplicate and eliminate any amplification bias that would occur during endpoint library amplification. The location of the UMI is in between our P5 adapter and our random primer. The way that the UMI is incorporated into QuantSeek is by replacing the second strand synthesis primer in the standard protocol. So importantly, this does not add any steps or any additional time to the protocol, since it is a one-to-one reagent substitution. The UMI itself is a short 6 base pair segment followed by a 4 base pair spacer. So the minimum recommended read length when using the UMI module is single read 75 chemistry on your sequencing instrument. Now, in addition to the UMI, QuantSeek is compatible with a number of additional products that we have, one of which is globin block. This is a globin blocking oligo to reduce reads that would be mapping to globin in your sequencing data when working with whole blood as your sample. We also have dual indices available for QuantSeek. We have 96I5 and 96I7 for 384 true unique dual indexing combinations. There's a targeted RNA-seq kit capable with QuantSeek, as well as FFPE compatibility for low quality samples as well. In general, QuantSeek is a very flexible protocol. We can handle quite low inputs of total RNA, and due to the low read depth required, is a very cost-effective way to get gene expression information and multiplex high numbers of samples. We partnered with Bluebee to enable really comprehensive data analytics capability with QuantSeek. This is free for customers of QuantSeek. The code is provided with the kit. It doesn't require any specialized bioinformatics knowledge. The data analysis itself is very quick, and Bluebee is a highly secure cloud-based platform. And recently we have released the UMI pipeline available on Bluebee for analyzing QuantSeek data that has been performed with the UMI module. So with that, I will hand it over to Andreas, who will talk more about Bluebee's capabilities, and thanks for your attention. Please add any questions into the chat box, and as Yekaterina mentioned, we'll be happy to answer those at the end of the presentation.
Thanks, Alex, for the first part. We are coming now to the second part, where we'll give you a short overview of how you can process the data, as Alex already mentioned. Here we go. For the downstream analysis, the bioinformatics part, we have developed together with Lexogen 3 different pipelines for the forward, for the reverse, and for the forward UMI kit. The pipelines are specifically configured, tailored, that they cover the differences between the library preparations between the 3 different kits. So you don't have to worry about how to configure it, which settings to choose, and everything is pre-configured and ready to use. And you don't have to think about how to do it and where to do it. What does easy to use mean? Together with the QuantSeek kit, you get an activation code from Lexogen that comes with the kit. You can use the activation code. You create your own account on the Bluebee platform. You are in full control of the account. The data stays in there, fully private. You control who has access to the account. You can invite additional users to register there. There is a pre-configured QuantSeek project inside the account that already contains all the pipelines that match to your kit. So if you buy a QuantSeek forward kit, you will see the forward pipelines in that project. After installing the connector for the data transfer, you can upload the data directly to your project. And as soon as the data arrives, you can easily select the files and start the analysis. As Alex already mentioned, that's very fast, short turnaround times. As soon as the analysis is done, you get a notification that's either by email or by short text message. And afterwards, you can easily download the data to your local environment. How does the pipeline look like, the workflow? So as mentioned before, there are 3 different pipelines for the forward, for the reverse, and for the forward UMI kits. The settings are optimized for each kit. You don't have to configure anything manually. If we look into the workflow of the pipelines in more detail, there is a generic outline of a few steps that's common to all pipelines. So there is an initial fast QC step, just to check how the quality of the input data is. That's using fast QC. After that, we are doing a trimming step to remove adapter sequences, low quality reads, poly A tails from the reads. And then the second fast QC step, just to see how the quality of the reads improved through the trimming step. Next step is the mapping step using star aligner. Here we, in the meantime, support more than 30 different genomes. So whatever it is, it's kind of likely that we have it already. If we do not have it, it can be easily added as an additional pipeline, so to support your research. After the mapping step, there is an RSEQ-C step that provides some statistics QC about the mapping step, mappability, uniquely mapped reads, numbers like that. And the last step is HT-sec count. Within the end, provide the main output, the main result of the pipeline, a list of genes with how many reads were mapped to those genes. So that's the main output you have at the end. One of the differences in configuration, for example, between forward and reverse pipelines, you can find here in that step, because strandiness of those 2 kits is different. So that's covered already in those 2 different pipelines that we have for the kids. With the new forward UMI kit that Lexogen released, we have 2 additional steps in the pipeline. So those are marked red on the right side in the overview. The first step in the beginning of the pipeline just removes the UMI sequence from the actual read sequence and adds to the label of the read. So the information is kept, but it's removed from the read for the processing. And after the mapping, you have an additional step that collapses the redundant reads identified by position and the UMI sequence. As an additional option, there is a pipeline for differential gene expression, where you can use the gene count files from the QuantSeek pipelines, use them as input for 2 different experimental conditions, and just start differential gene expression analysis using DESeq. Result files are an MA plot, a PCA, and a number of differentially expressed genes. All those files can be downloaded afterwards directly to your local infrastructure, so you can work there. They are kept for a limited time on Bluebee platform. All this is part of an integrated solution on the Bluebee platform. The analysis can be started automatically. So if you're doing this in high throughput, you can configure the connector for the data upload that immediately after the upload finishes, automatically starts the analysis. Then you get the notification and can download the results. If you don't want to automate it, but are working in high throughput, you can also select a batch of files and start the analysis manually, but for all of the files in one run. One other benefit of working on Bluebee platform is that you can share your results, your data with co-workers. You can easily invite additional users to your account. You can manage your data, your results in different projects. You have a kind of granular way to manage access rights to the project, and you can invite anyone from inside your organization, but also from outside. So just by sending an email with a registration link. There are different additional functionalities on Bluebee platform available. So if you want to process your data in a different direction, or if you want to get deeper insight in the greater overview of data you have, we are going to release Bluebees this month, which is a database that allows you to aggregate and mine your results. So if you have hundreds, thousands of expression profiles from individual experiments, you can easily upload the data and try to find some common patterns inside. That's it from my side, and I'm happy to hand over now to Siobhan. Thank you for your attention.
Thank you so much. This is Siobhan Gupta, and thank you all for giving me a chance to speak about my research today. So I'm a postdoctoral scholar at the University of California, Riverside, and I work with flame retardants, the specific flame retardant TDCIPP, and the focus of my research is to investigate the role of this particular flame retardant in altering the normal trajectory of zebrafish embryogenesis, and in turn look at their developmental toxicity. So flame retardants, these are chemicals that are used to prevent spreading of fire by chemical or endothermic reactions. And these kinds of compounds are found pretty much in a lot of items that we use in our everyday lives. As you can see, in kitchen appliances, in chairs and couches, in carpets, in baby items, in automobile, in car seats, in office furniture. So they're pretty much everywhere. And the particular flame retardant that I work with, TDCIPP, it's more specifically an organophosphate flame retardant, or OPFR. And these flame retardants have been widely used in the past 10 to 13 years, since another previous class of commonly used flame retardants, the PBDEs, were phased out in 2005 due to concerns of bioaccumulation and toxicity within the environment and humans. So these class of flame retardants, particularly these OPFRs, these are additive flame retardants, which means that they are not chemically bound to their end-use products, which gives them the potential to leach into the surroundings. And the routes of exposure, what happens is, like, the people living in those environments, they intake these flame retardants through inhalation of dust or exposure through skins. And there's been a couple of epidemiological studies coming out in the past years. This one particular study came out, which uh, which uh, which did a chemical analysis of urine samples from a representative US population, and they showed that 92% of those samples contained high detectable levels of BDCIPP, which is a metabolite of TDCIPP, the flame retardant that I work with. And in general, detectable levels of these OPFRs have been found in urine samples of children, breast milk of lactating mothers, placental samples in pregnant mothers, which kind of shows that they have a detectable neonatal or early developmental presence. And particularly these studies, again, these came out within the past 1 year, which shows a correlation between between TDCIPP exposures in the parents to different abnormal developmental outcomes. Like in this case, in the left in the paper on the left, they showed that there was a fertilization decline with increase in parental TDCIPP exposures in couples undergoing in vitro fertilization, whereas in the one in the right, they showed that this prenatal exposure to TDCIPP was associated with shorter gestation period. So all these kind of necessitate the importance of studying the impact of TDCIPP exposures from a developmental toxicity perspective. So, so in our lab, we use zebrafish as a model, and it's a robust model for studying developmental biology. It's a well-established model in the field of biology, and particularly for developmental biology, it's a very useful model because of their fast embryonic development. As you can see, right after fertilization, it takes only it takes only 24 to 48 hours for organogenesis within these embryos. So it's pretty. So the experiments that we do are pretty fast turnover. The embryos are transparent, so it enables us to view any developmental defects easily, very microscopically. And since this is a very well-established model, they have a well-annotated genome or transcriptome, and also a lot of the molecular biology tools are easily available. So using this zebrafish as a model, our lab has been studying the impacts of TDCIPP for the past around 8 to 10 years. The first paper that identified TDCIPP as a developmental toxicant was this 2012 paper, where it was identified from a screen of 4 chlorinated phosphate esters. And this paper, also showed that TDCIPP causes significant delays in remethylation of the genome. So basically, it also showed that TDCIPP has an epigenetic effect in the embryo. The next couple of studies within our lab were more focused on the epigenetics aspect. So in this 2016 study, the authors using whole genome bisulfite sequencing shows that TDCIPP exposures at around 0.75 hours post-fertilization, which is during the cleavage stage of the embryo, it predominantly led to genome-wide hypomethylation at 2 hours post-fertilization. And then uh, this study that came out last year, we showed that again this cleavage stage exposure of TDCIPP, it induces a delay in the process of epiboly. So if I can point this out, epiboly is the process in this developing embryo. The cells undergo cleavage, followed by the processes of blastulation and gastrulation. And during gastrulation, the cell mass, which was developing in the animal pole of the embryo, they descend around the yolk sac. And this whole process is called epiboly. And again, what we showed was that early exposures of TDCIPP induces a delay in the process of epiboly, and then such delay, consistent with the previous study, this delay was preceded by a hypo or hypermethylation of the genome. So based on these results, the overall objectives of my study were first to investigate or characterize the role of TDCIPP in inducing developmental deformities within the embryos. Based on that, I sought to assess transcriptomic changes associated with this developmental abnormality. So took more of a mechanistic path using mRNA-seq and reverse genetics, and then finally assess the downstream effects of this of the development of specific tissues and organs based on this transcriptomic data. So first I'll talk briefly about the phenotypic study, because that's what led to our the hypothesis that led to our transcriptomic aspect of the project. So again, similar to the previous studies, I started my exposures at 0.75 hours post-fertilization, which was during the cleavage stage of the embryo. And what we saw was that TDCIPP induces these epiboly defects. But in this case, what I saw was that these defects were of two kinds. A significant proportion of the embryos were epiboly delayed, which means they entered the process of epiboly, but they were delayed by half an hour or an hour. Whereas a large proportion of the embryos were epiboly-arrested, which means they did not enter the process of epiboly at all. So the cell masses didn't descend around the embryo at all. And there was a dose-dependent increase in the proportion of, in the strength of this phenotype. So at the higher dose or at the higher exposure concentration, more embryos were epiboly-arrested. Now, when these embryos were immersed in the exposure solution for 24 hours, what we saw was that the most of the embryos or most of the epiboly arrested embryos were dorsalized, which means if you can see in this panel, they had proper development in the head region, but they lacked proper development in the tail region. And as you can see, they're similar to the epiboly phenotype. There are different strengths of this dorsalization phenotype, all the way from class 1, where you can see there's only a bent tail, all the way to class 5, where the embryo doesn't even properly differentiate into heads and tails. So again, similar to the epiboly phenotype, there was a dose-dependent increase in the strength of dorsalization. But what was interesting was that these dorsalization phenotypes were similar to another well-known compound called dorsomorphin, or DMP as we call it here. And this DMP is a BMP pathway antagonist. So I'll talk about BMP pathway in a little bit, but it's an important pathway for development of it's an important signaling mechanism for development of an organism. And the embryos that were exposed to TDCIPP were phenotypically similar to embryos which were exposed to DMP, which led us to hypothesize that TDCIPP might also be acting through a BMP pathway antagonism. And these effects, when we started exposures at different times, 0.75, 2 hours post-fertilization, 3 hours post-fertilization, we saw that there was a strong shift in effects, a strong shift in the epiboly effects between exposures that were started between 2 and 3 hours post-fertilization, which shows that this is probably the window of sensitivity of TDCIPP-induced effects. And coincidentally, this is also when the process of cleavage ends and the process of blastula starts. So we established that the window of sensitivity of TDCIPP-induced effects was probably during the early blastula stage. Now, as I told you, BMP signaling, it's an important signaling mechanism for embryonic development, particularly in zebrafish embryos during the process of gastrulation. There's a ventral to dorsal gradient of BMP signaling. So in the ventral region, the signaling is high, whereas in the dorsal region of the cell mass, the signaling, there's almost no BMP signaling. And as you can see here, this is reflected this is reflected using immunofluorescence in this particular embryo. This is an embryo stained with the phosphoSMAD 159 antibody, which reflects the ventral to dorsal gradient of the BMP signaling. And this, due to this gradient, the BMP signaling helps in the development of the ventral structures like TEL. So a BMP inhibition, similar to what we saw in dorsomorphin exposed embryos, leads to these dorsalized embryos which lack a development in the ventral region. Whereas a BMP signaling overexpression leads to ventralized embryos, where we see an overdevelopment in the tail region and a lack in development of the head region. So again, based on our phenotypic study, we saw that TDCIPP exposed embryos phenocopy dorsomorphin exposed embryos. So our hypothesis was probably TDCIPP acts through BMP signaling inhibition. So we used a BMP signaling activator or a BMP agonist to see whether it rescues TDCIPP induced effects. So we pre-treated the embryos from 0.75 to 2 hours post fertilization to this compound called 4' hydroxychalcone, which is a BMP agonist, and then continued on with TDCIPP exposure till 6 hours post fertilization. And what we saw was, if you look at these graphs, what we saw was basically that a 4' hydroxychalcone pre-exposure led to a partial but a significant rescue of this TDCIPP-induced epiboly. Now, these led us to this like strengthened our hypothesis that most possibly TDCIPP was acting through a BMP signaling inhibition. So to verify that, we use transcriptomics as a tool. So we did transcriptomic studies over different stages using mRNA-seq and reverse genetics that I'll come to in a while, with a focus on BMP signaling. So again, coming back to this cycle, we started our exposures at 0.75 HPF, and uh, and we collected samples all between 3 and 24 HPF. So we collected at 3, 4, 5, 6, and then 8, 10, 12, and 24 HPF, which kind of led to around 90 samples. We did 4 exposure concentrations. The 0.1% DMSO was our control. We did 2 TDCIPP exposure, 2 TDCIPP doses, and 1 dorsomorphin dose as a positive control. And these led to 90 samples across 8 time points. And this is where the Lexogen Quantseq forward came in, because we had a lot of samples and we were looking for a library prep kit, or we were looking for a library prep uh, uh, library prep mechanism where we would not which would be cost effective, and we could do it in a limited time and within a limited cost. And particularly what also drove this was that we had recently bought a Miniseq in our lab, and the Lexogen Quantseq forward kit at that time was the only whole transcriptome kit or full transcriptome kit that supported a Miniseq. So we used the Lexogen Quantseq library prep kit and then did a whole transcriptome sequencing using our Miniseq. We used the Miniseq High Output kit, and uh, what also was important here is the kind of the uh, is the partnership between Lexogen and Bluebee. So I'm not a hardcore bioinformatician, so I needed a streamlined bioinformatics platform, and the link between the Lexogen and the Bluebee. So what all I had to do is we use BaseSpace for the sequencing. So all I had to do is download the FASTQ files and upload it to the Bluebee pipeline, and then using the Bluebee streamlined pipeline, the alignment and the differential gene expression was automatically done. And the another important fact was that this Bluebee pipeline consisted of the zebrafish GRC10 database, which is one of the most updated databases of the zebrafish genome, which was lacking in at that time, and I think it's still lacking in most of the established pipelines. So we did the alignment, we did a differential gene expression, and based on the results, we did a gene ontology pathway using we used like two different two different applications. We used David, and then we used IPF. So this is basically to show, so we used 8 flow cells, 1 flow cell for each time point, and what is shown here is basically using the Lexogen library prep. We got pretty robust Q30 percentages, so we got like really good quality data and sequencing runs out of these libraries. And these are a subset of the time points that I sequence. As you can see here, the earlier time points did not have a lot of differentially expressed genes. The red ones are the ones embryos that are exposed to TDCIPP, whereas the blue ones are the ones that are exposed to dorsomorphin. And the earlier time points did not have a lot of differentially expressed genes, and this is consistent with one of our previous studies where we don't see a lot of sensitivity at earlier time points, but at 24 hours we saw a good amount of differentially expressed genes. So as I said, our hypothesis was to see whether TDCIPP has a direct effect on BMP signaling. So we isolated from our sequencing data, we isolated our BMP signaling transcripts to see whether there was a overall TDCIPP-induced effects. But surprisingly, we did not see, as you can see here from the transcriptomic data, we did not see any impacts of TDCIPP and BMP signaling across the different time points. Whereas dorsomorphin led to the inhibition of some of these transcripts, which is consistent with its established mechanism. To confirm that, we again used a phosphosmand 159 immunofluorescence, like I showed earlier. And in a normal embryo, it shows a ventral to dorsal gradient of BMP signaling. And this gradient was still maintained in the TDCIPP treated embryos. Whereas for the dorsomorphin treated embryos, this gradient was disrupted, which shows a BMP signaling disruption. The one transcript that was significantly downregulated in all our treatments, in both TDCIPP and dorsomorphin treatments, surprisingly, is this gene called sizzled. The sizzle gene, it's a part of the BMP signaling mechanism. So a downregulation of sizzle, we initially thought that all our TDCIPP induced effects might be driven by the sizzle downregulation. So to test this, we designed a splice blocking morpholino targeting the sizzle gene and microinjected the embryos with this morpholino. But what we saw was that the sizzled morphans at 6 hours post-fertilization were completely normal. And at 24 hours post-fertilization, the sizzled morphans were ventralized instead of dorsalized, which shows that although sizzled is downregulated in TDCIPP treatments, this necessarily did not transfer, or this necessarily did not translate to a specific phenotype. So then the open question was still, how does TDCIPP induce all these developmental defects? So we did a gene ontology analysis using data we got from 10 to 12 hours post fertilization sequencing. And what we saw was a number of these pathways were affected by TDCIPP. But what I'm going to talk about today is its impact on mesoderm development. So in a gastrulating embryo, as I said, the embryos the cell mass descends around the yolk sac, and the cell mass arranges into the three germ layers: the ectoderm, mesoderm, and the mesendoderm. Particularly for the mesoderm, and I'm going to focus on that today. This mesoderm, this remains in an undifferentiated state early during the gastrulation, and this undifferentiated state is maintained by the Wnt signaling. Now, during gastrulation, as gastrulation proceeds, these mesodermal cells, or the MPCs, or the mesodermal progenitor cells, they start differentiating under the regulation of these transcription factors, mesogenin 1 and TBOX16. And ultimately, these form the presomatic mesoderm, or the PSM, that basically downstream differentiates into different tissues and organs like the somites, the notochord, the heart, and the blood. And we'll talk about it. We'll talk about all this soon. Now, during this process of gastrulation and during the stages following gastrulation, if we stain the embryos with the TBOC16 antibody, as is done here, we'll see that the mesodermal cells are localized towards the tail bud of the embryo. But when we looked at the sequencing data and isolated these transcripts, what we saw was that TDCIPP led to a downregulation of this like mesoderm specific transcripts like TBOC16, TBOC6, and mesogenin-1, which dorsomorphin did not. So this shows that TDCIPP might be acting through a direct impact on mesodermal integrity. When we stained these embryos with the TBOC16 antibody, what we saw was that unlike the normal embryos, where I just pointed out now that the mesodermal cells are localized around the tail bud, in the TDCIPP treated embryos, the mesodermal cells are delocalized. So there's no specific localization of no specific localization of the TBOC16 staining, which again shows that TDCIPP most possibly has a direct impact on mesodermal integrity. And probably, although TDCIPP and dorsomorphin have the same phenotypes, they might be acting through different mechanisms. So based on this, my next objective was to assess downstream effects on development of the specific tissues and organs based on this altered transcriptome. And I basically focus on studying the tissues and organs that the mesoderm differentiates into. So first, I looked at these structures, the somites and the notochord. Now, again, like during somatogenesis, which is the period following gastrulation, a normal embryo has a well-developed notochord, and the somites radiating out of the somites radiating out from the notochord. But again, in a TDCIPP treated embryos, we did not see any evidence of a well-developed somite or a notochord. We did a gene ontology, since at 24-hour or 24 HPF, we had a lot of differentially expressed genes. We did a gene ontology based on that specific time points. And what we saw was that consistent to a mesodermal disruption, a couple of pathways which are related to cardiac development as well as oxygen transport, which is in turn dependent on blood cell development. These pathways were affected by TDCIPP exposures. So first, we looked at red blood cell development. We use this staining technique called the o-dianisidine staining, which stains hemoglobin. And what we saw was that TDCIPP treated embryos, by 72 hours post-fertilization downstream in development, these embryos are largely anemic. Consistent with this, we saw from the 24-hour data, we saw a subset of transcripts that are related to hemoglobin synthesis and maintenance. These transcripts were downregulated. So this shows that as a downstream function of mesodermal disruption, there is a direct impact on blood cell development or erythropoiesis. Now, since mesoderm also differentiates into the cardiac tissues, we looked at cardiac development. And again, what we saw here was that TDCIPP leads to a direct an increase in the pericardial area, and this is considered as a marker of cardiac defects. And as you can see here from these images, a lot of these TDCIPP in treated embryos had defects in cardiac looping, which means that their hearts didn't loop properly during the cardiac development. And almost like 40% of the embryos show this cardiac looping defect. And again, consistent with this, at 24 hours post-fertilization, we saw a decrease in transcript levels of the specific genes that regulate cardiac development. So all this, the absence of a proper development of a notochord or a somite, the induction of anemia, and the impact of cardiac development, these are all fuel from the mesodermal disruption we saw earlier. That is in turn fuel from fuel from the epiboly defects that we saw. So again, just to summarize, when we expose the embryos to TDCIPP at 0.75 hours post-fertilization around here, around 6 hours post-fertilization, we see these epiboly defects, but we don't see any large-scale impacts on BMP signaling. We see mesodermal disruption. These epiboly defects ultimately lead to mesodermal disruption. We see a dorsalization of the embryos, and then downstream during the development, we see a defect in the mesoderm-derived structures or organs or tissues, like the heart, like the heart or the red blood cell formation. So, as I said, using Lexogen and Bluebee helped a lot because it was cost-effective. We could do the library prep for our 90 samples within I think $2,000 to $2,500, whereas we kind of checked a lot of other library prep kit, and a lot of them cost like almost double the price of what we got with Lexogen. It supports Miniseq, which was a big deciding factor at that time. They have a streamlined bioinformatics with Bluebee, and particularly the availability of the zebrafish, required zebrafish genome, one of the most updated zebrafish genomes within Bluebee. From the sequencing data, we got a good consistency between the phenotypes and the differentially differentially expressed genes. And particularly one of the things which we have been most satisfied using Lexogen and Bluebee is their efficient scientific and customer support. So I usually have, like when I do a library prep or when I do a new method, I usually have a lot of questions, and I email them, and they usually reply within like 24 hours, and it's like a pretty efficient reply. So we were like really satisfied with that. So just the acknowledgements, my mentor, Dr. David Woles, all my lab mates, Ali, this is Ali, this is Constance, this is Sarah, and this is Vanessa. And my project was funded by NIH, and this whole study that I talked about today was recently published in this paper, but there's also been a couple of papers that came out of our lab recently, this Mitchell et al. paper and the Vliet et al. Paper, which also used extensive levels of QuantSeek Bluebee sequencing. So with that, thank you for listening, and I'll take any questions.
Thank you everyone for these informative presentations. We really enjoyed them. And before we get started with our question and answer session, I would like to remind our audience how to submit questions. You can do it by typing them in questions box, which can be found on the right side of the presentation window. We'll try to answer as many questions as we can. We have 10 minutes, so for sure there will be several questions answered live, and if there are some questions left, we will follow up after the webinar. So I see there is a question which probably goes to Alex Moiher, and this question about how does UMI incorporation change the recommended read length and read depth? Could you please comment on this?
Yep. Hi. So the UMI incorporation doesn't necessarily change the recommended read depth. For gene expression, we recommend 3 to 5 million reads per sample. However, for read length, when using the UMIs, we recommend a minimum of single read 75 chemistry, or at most single read 100 for read length.
Okay. And then, thank you very much, Alex. There is another question which probably goes to Andreas. It's, can Bluebee be used for chicken samples?
Yeah, it can. We recently added the chicken genome, so it's supported.
Mm-hmm. Thank you for your answer. Another question goes also to Alex Moyher. Can we get 8 million reads from very low input RNA as 10 nanograms per microliter without overamplification?
Absolutely, you can get 8 million reads from 10 nanograms input. For QuantSeek, 10 nanograms is not necessarily considered low input. You can go lower than that, and we have protocol modifications available for using down to the 500 picogram level of RNA input. So 8 million reads from 10 nanograms is is very doable, particularly if this is a high quality RNA, with a RIN value of over 8 or above, or 7 or above. That's completely doable. I would also just like to quickly comment on the previous question to Andreas about the chicken genome. If you have a need for a genome to be put into Bluebee that is not there, you can write to Lexogen, and our bioinformaticians can upload a genome into the Bluebee, into our pipeline on Bluebee, so that you can run something that isn't there as well.
Thank you, Alex. And then a question to Subham. Did you experiment with the higher read depth to identify low expressed genes?
We did not. We did not specifically target for low expressed genes. So we just targeted for a differential gene expression to identify pathways, but we were not looking at specific low read genes. So a lot of the low read count genes were probably like filtered out during the filtering process.
Thank you. There is a question, what normalization procedure should be used, quantile normalization or other? So maybe Alex or Andres could comment on this question, because it's related to data analysis.
Yeah, discussions about normalization are usually a little bit difficult. Depends very much on the experimental setup that you have, and how your data looks like, and what you want to achieve at the end. There are different options. They all have their advantages and disadvantages. So there is no generic rule how to do it.
Mm-hmm.
As far as I know, Blue probably uses the DESeq based, the variant stabilizing normalization. That's what
That's true. Yeah.
Okay, thank you. And then there are two consequent questions, and Alex, I can see, has already replied in written to one of these questions, but maybe you can repeat, and then we come to the second one. So the question is, how many reads do you recommend per sample using QuantSeek? And what is your view on biological replicates versus technical replicates? What are your recommendations regarding this?
Sure. Yeah. So the recommended reads per sample for gene expression is 3 to 5 million reads. With biological replicates, those can show variance, so we recommend technical replicates, and in a perfect world, 3 technical replicates would be ideal. And just another comment on the previous question about the normalization. DESeq is the default normalization that we use in the Bluebee pipeline.
Thank you, Alex. And then the second question, which comes out of this question, how should samples below 3 million reads be interpreted? Do another technical replicate?
This will kind of depend on the situation that you have and the sample quality. I mean, there are people that go less than 3 million reads per sample and still are seeing good gene expression data. So this is kind of a hard question to give a specific answer. You can certainly do another technical replicate, but it's not necessarily that less than 3 million reads per sample will not give you usable data for gene expression.
I'd like to step in here that a lot of my sequencing runs had like less than like 3 million reads, and most of them give like pretty good data. So I don't think that's a rate limiting thing here.
Okay, we have time for one more question, and actually there are quite few, and we will go for one of them, and then the rest we will reply via email. So the question is whether do you provide the settings of the pipeline? And question is for Andreas.
We do. They are not exposed directly on the platform, but they are described in detail in the user guide. So you can get all the information, how the individual steps are executed, how the settings are from the user guide.
Thank you. So yeah, thank you very much. I would like to once again thank all the speakers for their interesting presentation, and for all the audience for your information. Recording of today's webinar will be available, and you will receive an email with the link for download. And we invite you to forward this recording to your colleagues who might have missed today's live event but would like to hear about QuantSeek and Bluebee and research of Subham. Yeah, and we will follow up with the questions which were still left out. Thank you very much.